Hello guys, welcome back. So as you know, we are in the last part of the cell cycle control system. If you saw the previous videos, we are doing the cell cycle. So in the last part we studied about the mitosis 5 stages, right? Prophase, prometaphase, metaphase, anaphase, and telophase. Right? So the last phase of the cell cycle is M phase. It consists of mitosis and cytokinesis. Correct? So we studied mitosis. Now the last thing is cytokinesis. Right? So the last thing, I will just write it down here. Cytokinesis. That will see. It's just fairly simple. So the cytokinesis is the process by which the cytoplasm is cleaved into. So it cleaved means it separates into two. That completes the end phase, right? These are the cells as we saw in the telophase, right? So like the telophase is something like this, the cell shape something like this, and it will have two separate daughter nuclei, right? And the cytokinesis process is just it have to break into two, right? So the cytokinesis process usually begins in the anaphase, right? But it is not completed until the two daughter nuclei have formed in telophase. Whereas mitosis depends on, right? The entire mitosis depend on the transient microtubules that we studied here. This microtubules base structure, the mitotic spindle. So the mitosis entirely depends on the microtubules length. This is how it separates, right? The cells. Whereas the cytokinesis in animal cells depends on transient structure based on actin and myosin filament. So the cytokinesis depends on actin and myosin filaments. So this depends on actin and myosin filaments that make this two makes the contractile ring. What it makes? This makes contractile ring. Alright, so both the plane of cleavage, right, the plane of this where it has been separated, and the timing of cytokinesis, however, are determined by the mitotic spindle. So both of this is determined by the mitotic spindle. So the mitotic spindle, right, decides where the plane where it will be separated or where the furrow will be developed. So first visible sign of cytokinesis in an animal cell is a puckering and furrowing of the plasma membrane that occurs during the anaphase. Right, so that you are seeing a picture. So that's an electron micrograph of a frog cell, and it shows the line you are seeing. It's the furrowing of the cytokinesis. So the furrowing invariably occurs in a plane that runs perpendicular to the long axis of the mitotic spindle. So this positioning ensures that the cleavage furrow cuts between the two groups of the segregated chromosomes, right? So that each daughter cells receive an identical copy identical and complete set of chromosomes. So if the mitotic spindle is deliberately displaced, right, as soon as the furrow appears, if the furrow appears and if the mitotic spindle is displaced, the furrow disappears again and a new one develops at the site corresponding to the new spindle location and the orientation, right? So totally depends on the mitotic spindle. And once the furrowing process is well underway, however, cleavage proceeds even if the mitotic spindle is artificially sucked out of the cell membrane, of the cell, or depolymerized using the drug called colchicine, right? So these are some of the possibilities. So even if you shut the mitotic spindle outside after the furrowing is developed or furrowing is processed, then it will still process, however, if there is no mitotic spindle. So the mechanism is still not so clear about how the mitotic spindle dictate the position of the cleavage furrow. But it appears that during anaphase, the overlapping interpolar microtubules. You remember the overlapping interpolar microtubules, right? So these are our two uh, mitotic spindle, right? So the overlapping means overlapping interpolar microtubules means this is the one are called interpolar overlapping interpolar microtubules, right? That forms central spindle. So this form the central spindle recruit. So this interpolar microtubules, what it does, they believe that it recruit and activate proteins that signal to the cell cortex to initiate the assembly of the contractile ring at a position midway between, so exactly at the center of this thing. So something like this, it develops a contractile ring at the center of the mitotic spindle. And because this signal originates in the anaphase spindle, this mechanism also contributes to the timing of cytokinesis in the late mitosis. And when the mitotic spindle is located centrally in the cell, the usual situation in the most dividing cell, the two daughter cells produced will be of equal size. Right, and during the embryonic development, however, there are some instances in which dividing cell moves its mitotic spindle to an asymmetrical position, and consequently the furrow creates the two daughter cells that differs in size. So sometimes the daughter cells are differing in size because the mitotic spindle moves their position. In most of these asymmetric divisions, the daughter also differ in the molecules they inherit, and they usually develop into different cell types, right? So the contractile ring is developed only in the animal cell, not in the plant cells, and it is made up of actin and myosin filament, right? Right, so the contractile ring, right, it's composed of an overlapping actin and myosin filament. So it's something like this. So for example, this is the contractile ring. So it will have like actin filament and myosin filament and actin filament and myosin filament. This is actually around this, right, like this, right? So it disassembles at anaphase, and it is attached to the membrane associated proteins on the cytoplasmic face of the plasma membrane. So once it is assembled, the contractile ring is capable of exerting a force strong enough to bend a fine glass needle inserted into the cell before cytokinesis. So much of this force, how this force is generated by the sliding of actin and myosin filaments against each other. And unlike the stable association of actin and myosin filament in the muscle fiber, but however, in this position, the contractile ring is just a transient structure, and it assembles to carry out cytokinesis, gradually becomes smaller as cytokinesis progress, and disassembles completely once the cell has been cleaved into two. So it grows like this, like this, like this, and it separates the cell. Right, so cell division in many animal cell is accompanied by large changes in cell shape and a decrease in the adherence of the cell to its neighbors, to the extracellular matrix, or to both. So this changes results in the part from the reorganization of actin and myosin filaments in the cell cortex, only one aspect of which is the assembly of the contractile ring. Right? So that was our cytokinesis process. So that's just a simple cytokinesis process. But and so this is just happens in the animal cells. Like in the plant cells, it's a different process. The cytokinesis actually built a cell wall, not a contractile ring. So that's, if you're studying botany, you can go more in details, but I'm not going into the cytokinesis of the plant cell. But this is how in the animal cells that we need to know. This is how it is formed, right? All right, guys. So that completes our entire cell cycle. So we studied the cell cycle from the G1 phase as phase G2 phase and M phase. We studied the cell cycle control system, how the mechanism that control this entire cell cycle. Alright guys, so like this is a very long process, right? But I will make a summary of this entire cell cycle with using the most important points, and maybe that will help you just to revise when you're revising the cell cycle. Alright guys, uh, thank you guys for watching, and we'll see you in the next